Hello, I'm Bohdan Schneider. I coordinate activity on nucleic acids in 3D Bioinfo, Elixir community. I work at Institute of Biotechnology of the Czech Academy of Sciences, and we will have this webinar will split into 3 relatively short talks, but I hope they will be interesting for you. At the beginning, we will concentrate on RNA, which is probably attracts more interest recently, especially from the 3D point of view. Modeling of 3D structures of RNA is still a challenge. And there will be a first talk by Marta Szachniuk from Poznan, from Poland, on one particular method, how to work with 3D RNA modeling, or how to approach it. And then we will hear from Zhichao Miao from EMBL-EBI and from Chinese university, which I'm sorry, I don't know. You can tell us what it is, because you also have Chinese affiliation, as I understand. And Zhichao works with Eric Westhoff on organizing really important activity that's called RNA Puzzles, which is which concentrates people who try to model RNA or predict RNA 3D models. And this competition runs for like 10 years now. So there will be some report about the rules, I guess, and results, recent results. And the last talk will be by Jiří Černý from Institute of Biotechnology, the same institute as I am in, Czech Academy of Sciences. And Jiří will talk about some tools we are developing here that are aimed at annotation, validation, and refinement of nucleic acid structures, both DNA and RNA. So I think we should proceed to the actual talks. So it's Marta, it's all yours.
Okay, thank you, Bogdan. Good afternoon, everybody. So my talk will be about interlaces and lassos. That are 2 topologies that appear in RNA structures. I will talk about what they are, where and how we find them, and what comes out of it. And the story begins actually with RNA Composer. That is our system for RNA 3D structure prediction. In my lab, we've been developing this system for about 10 years now, and we are constantly trying to improve it, to improve the quality and accuracy of predicted structures. And to do it, we closely look at the output of RNA Composer. We analyze the results, but not just the final results, but also the results of intermediate steps of computing. And through this analysis, we identified the problem of entanglements. And it turned out that from time to time, the system built models with entangled structure fragments. Most of models with entanglements are rejected in the final processing stage. So we do not get them in the output. However, a few times it happened that they slipped through the internal evaluation. And so we noticed the problem and we got interested in it. So we decided to take a closer look at it. So let's start from some definitions to clarify what we mean by entanglement of structural elements. The starting point here is the secondary structure, although we talk about entanglements in the tertiary structures. So in the secondary structure, we can distinguish different elements of the structure, like loops, different types of loops, like apical, internal, multi-branch loops, badges. We also can see stamps, dangling ends, and so on. What we do is we divide these elements into two groups. So we say that we have open structure elements and closed structure elements. Open structure element is the one that is not closed by any base pair. In this figure, you see here all these orange things just are open structure elements. And closed structure elements are loops, different types of loops, and dinucleotide steps, that is parts of stems. Now, when do we have the entanglement? If in a structure, an area inside a closed structure element is punctured by another element, then we have entanglement. And theoretically, we can have the following types of entanglements. In general, we can divide them into two types, interlaces and lassos. So interlace is when we have two closed elements of the structure, and each of them is threaded through the other one, through the area of the other one. And a lasso, we have a lasso when one element, one closed structure element embraces the other element of any type. To characterize the entanglements in structure, we introduced some notation. You can see it here. So we just use 3 letters to denote loops, nucleotide step, and single strands. And we use ampersand and brackets to denote if we have interlace or lasso type entanglement. Okay. And what we did next, we decided to screen all the predicted models, the models predicted within RNA Puzzles, to find out whether these models include entanglements or not. So whether it is a more general problem or just a problem that our system has. And last year we published a paper about with some toolkit, and we also published a benchmark data set. The benchmark includes all the data coming from 22 RNA Puzzles, RNA Puzzles challenges. So the repository includes 22 challenges, the data from 22 challenges, and we have 22 target structures there. There are express structures and over 1,000 predicted RNA 3D models that were submitted to these 22 challenges. What we did with the tests, what this benchmark said, we decided to remove all the redundancy. So after removing redundant models, we were left with 995 non-redundant models, predicted models. 226 were predictions in web server category, and over 700 were predictions in human category. And we screened all these models for entanglements. What we found was that 14% of all these models included entanglements. Mostly they included only one entanglement, but we had also models with 2 to 5 entanglements per model. In all these models, there were 138 models altogether with entanglements. We found 201 entanglements of different types. The majority were lassos. So we had 4 types of lassos found in these models. 2 types were not found. These were lassos of type DS and DL. That means that we didn't find lassos where the dinucleotide step embraces single strand or loop. And we found all 3 types of interlaces. It is 18% of all the entanglements found in the predicted models. Then we tried to analyze the relationship between entanglements and measures or parameters of structures. What we found is that there is no correlation between RMSD and entanglements. We also did not find any correlation between clash score or molecule size and entanglement. But what we found is that entanglements are strongly correlated with pseudoknots. So in general, the probability of entanglement in pseudoknoted RNA is 4 times higher than in case of non-pseudoknoted structure. And probably the systems that generate predictions, they are somehow fooled by pseudoknots and try to make a structure, and instead pseudoknots, they simply generate something with entanglements. Okay, then we asked if there are any methods that do not predict entangled structures, and the answer is no. All of methods happen to generate entangled structures. We tried to formulate some hypotheses why or when these entanglements may appear, and what we think is that in fragment assembly-based methods, entanglements appear during the assembly stage, and in de novo methods that simulate coarse-grained models, entanglements may appear during the reconstruction to the full atomic model. Okay. Next, we decided to look if there are any entanglements in experimental structures. And so we went to Protein Data Bank and we downloaded all the RNAs that we found there. It was at the beginning of August this year. So there was a little more than 5,000 RNA structures. And we screened all of them for the occurrence of entanglements. And what we found is that also there are some structures with entanglements. Namely, there were 18% of structures, including entanglements. And we found all types of entanglements there, but just 0.5% were interlaces, and all the remaining entanglements were lasso type entanglements. Okay. What we also found in these data, we did not look deeply into them, but what we also found is that these entanglements we found in the experimental structures were mainly in the structures with poor resolution. Okay, and now how do we find entanglements? Of course, we do not do it manually. We have the algorithm that we developed. Now we've prepared a web server for identifying entanglements in RNA structures. The web server is named RNA Spider. It's already available on the web. So what we can do there is we can upload the structure or just give PDB ID, and the system analyzes the structure, and if it identifies an entanglement, it gives a list of entanglements, and it gives the visualization of the structure, the secondary and the three-dimensional structure, and it shows where the entanglement was found. Okay, so what are the take-home messages from this talk? So first is that the entanglements of structure elements occur in both the experimental and predicted RNA 3D structure models. We think that possible reasons of their occurrence there is poor resolution in case of experimental structures, and in case of predicted models, it's either improper fragment assembly or it's quite, let's say, careless conversion from coarse-grained model to atomic structure. And the third thing to take home is that we can identify entanglements automatically. We can use it. We can use our RNA Spider web server for it, or we also have just computing scripts without any user interface. They are also available to the people. Okay, and there is just a list of people who worked in the project. So it's Maciej Antczak, Mariusz Popenda, Joanna Sarzyńska, and Tomasz Żok. And the project was founded by two institutes from Poznan University of Technology and Institute of Bioorganic Chemistry of Polish Academy of Sciences, and also by the grant from National Science Center. Okay, thank you.
Thank you for perfect timing and for interesting talk. We have perfectly 5 minutes for questions, if there are any. You may ask.
I think Zhichao has posted something in the
Okay,
chat.
yeah, okay, I'm trying to.
Okay, I can see the question.
Yeah, you can see. Okay, so yeah, okay.
Do you have any suggestion how we can relax the structures with entanglements? At this moment, I don't. So at this moment, I don't have the remedy how to avoid entanglements. I can only identify them. Okay, so I think some relaxation is the next step, and we are thinking about that, but yet I don't know how to do it exactly.
So maybe you, I'm sure it was in your talk. So how many you observe in experimental structures? It's probably in larger structures quite common, if I understood.
Yeah, it was 18%.
Yes, it's a lot
of entanglement of structures included. So it's not a lot. They're mostly with poor resolution, but also, well, there are some problems with our algorithm in processing structures which are incomplete or have some incontinuous chains. So that's a problem because, of course, what we do when we find entanglements, we try to smooth the backbone. And if the backbone is not continuous or there are some quite large lengths of bonds, then we have problems. But anyway, I think that's the problem of entanglements in these experimental structures is the problem of people who are trying to compute these structures from experimental data. Yes, or let's say algorithms which are not that correct if you would like them to be.
And do you think it can never actually occur? Because some, topologically, yes, it would be probably very complicated, but I can imagine that RNA strand can go through some large loop, at least theoretically. But you think it's always a mistake?
No, I don't think so. I mean, it's now it's also one of the topic that is explored by not just us, but also by the people who are dealing with knots and structures. So some people think that maybe lassos can have some functionality. We don't know yet what kind of functionality, but probably they do. But I think that the second type, this interlaces, yes, this is, we can say that from topological point of view, mathematical point of view, interlaces like a hop link, yes, so there's something like that, okay? So these ones are probably incorrect. And as for lassos, some may be correct, some not. For example, I don't think that if the single strand is going through the nucleotide step, it's correct. Okay, so I don't think so. Probably this is incorrect. But if we have a single strand going through a big loop, probably it's okay.
Okay, thank you. So, is there any other question? If not, I would stick to the timing, which is still very, very good.
Okay, thank you.
So thank you very much, and I hope we can continue with the discussions, because I think we may have some tools that might even help you to improve your methods. But okay, that's sorry. So okay, Zhichao, RNA Puzzles. Please go ahead.
Oh, hello everyone. It's a great pleasure for me to present you RNA Puzzles, and thank you, Bohdan, for the invitation. So, Marta has already introduced you something about the problems in the prediction. So here I'm trying to introduce you something about um, the prediction game, which is RNA Puzzles. We try to assess how the prediction can work. So I'm Zhichao Miao from European Bioinformatics Institute. Uh, so before prediction, so one key point we need to keep in mind is that for any of the biomolecules, we assume that normally the sequence leads to one unique structure. This is the assumption for all types of structure prediction. And then probably in terms of structure prediction, you probably already know like the uh, protein structure prediction, like AlphaFold, which achieves a great success in the prediction. And so people also consider to some extent, the protein structure prediction has been solved. And nowadays, more and more people focus on the RNA 3D structure prediction. And but let's date back to the 1960s. We also had some early pioneering work about RNA structural prediction, and we know that this was also a mark of the beginning of bioinformatics, and especially the modeling of tRNA structures. And uh, what's the specialty for the RNA structure? So RNA structure has it has um, 2' hydroxyl, and also if we consider the base of RNA as a triangle, it will have three edges: the hosting edge, the Watson Crick edge, and the sugar edge. So if we consider this base of a RNA nucleotide as a triangle, and then the interaction of these three edges can be a combination. So there will be 12 different types of combinations in the interaction. And all of these types of interaction will form hydrogen bonds, and which makes the structure stable. But normally, when we say a base pair, means that we only consider the interaction between Watson Crick and the Watson Crick edge, and so the interaction is size conformation, and so it's only one type of the 12. But actually, we can have multiple interaction types, and then these different types of interactions will form some stable structures, and these structures are recurrent in RNA, and we call them as structural modules. These structural modules are formed basically they are formed with non-Watson Crick base pairs and embedded between the Watson Crick base pairs, which makes the structure stable. And these are recurrent, and so they are quite stable. So it can be difficult to predict these modules. And so RNA Puzzles RNA Puzzles is a collective experiment for the blind RNA structure prediction. So this is similar to the CASP in protein structure prediction. So we collect the sequence and then distribute the sequence to all the prediction groups, like different prediction methods, like the RNA composer, like FAFA or like VFold, all these prediction groups, and then these prediction groups try to predict the structures, and finally we compare the predicted structure with experimental determining the structures. So for the sequence, we already have the structure determined by a crystallography group. So here are some milestones in RNA Puzzles. So the idea started more than 10 years ago, and in the past decade, we summarized the 4 rounds of predictions. We also hosted 2 community meetings, and now we are not only having regular predictions, but we also organize some new puzzles. And for the COVID, we predicted some parts of the COVID RNA structure. And for the aim of the RNA Puzzles, basically, we want to determine the capability and the limitations of the prediction, And also we want to find if we have made a progress in the prediction, and we want to identify which are the bottlenecks, and if we can conquer these bottlenecks. We want to solve real-world problems using these predictions. For example, if we can solve the COVID problem, we also encourage to have some to develop some automatic prediction tools, so all people can benefit from this. So, and finally, we also want to explore some biology mechanism. And so, when organizing these RNA puzzles, we demand the confidentiality, so we need to protect the crystallographers. So crystallographers normally provide their sequence to us, and then we disseminate the sequence to all the prediction groups. So we need to keep all the information very safe, and the people need to, all the prediction groups need to sign the confidential agreement. They will not distribute the sequence information outside the prediction group. And also, we also only publish the modeling results after the X-ray publication. And so normally what we do is that we recruit the structure from the experimentalist, and then we give some days for preparation, and then for the prediction. There are two types of predictions. For the automatic web servers, they have 2 days for the prediction, and for human experts, normally it's 3 weeks or 4 weeks. And sometimes there will be additional experimental data available for the prediction. And then for the submission, each prediction group or each prediction method can submit up to 5 models. So they can be very different. And afterwards, we do the assessment or the evaluation of this prediction, and we summarize and release all the predictions. And here is the website of RNA Puzzles. And so you can go to rnapuzzles.org, and you can see all the previous puzzles and also the rules of our RNA puzzles, and you can see the comparison between the predicted structure and the X-ray structures. So for the one key point here in this RNA puzzles is that we need to do the assessment or the evaluation of the predictions. So we developed this RNA puzzle toolkit, which is a bundle of tools and can calculate different types of assessment metrics. Like we know the root mean square deviation, which is quite normally used, and also we developed the interaction network fidelity and the deformation profile. These are 2 metrics specifically designed for RNA, and also we have clash score, which was mentioned by Marta. This is the workflow, and also RNA tools is a very powerful tool, which is inside the RNA puzzle toolkit. Maybe you can have a look at it if you want to do some dealing with some RNA structural things. And for the prediction results, now we already have organized the 33 regular puzzles, and this is a summary table here. I do not dig into too much details, and this is the brief result about the round 4 assessment of the prediction. So we do see some improvements over the last 10 years, and we see some of the there is still some problems, like the non-Watson-Crick interactions is still very difficult to predict, and also some of the models still have very high clash score. So also Marta mentioned about this, and for this, this is a graphical view about our predictive structure, and we do see some of the prediction have been very, very accurate, and then the predictive model is similar to the experimental structure. And also here are more models here. So many of them belong to the AFAN families, and then we are thinking about that we can take advantage of these good predictions and to apply to real biological problems, like some of the AFAN family RNAs. So here is an example telling us that the 2D structure is not enough and the 3D structure is more powerful. And so so we find that one of the prediction has a 100% correct in the 2D structure, and but it's still far away in 3D. So we think 3D is more informative. And also, this is about the structural module prediction. For structural modules, we do find some of the predictions are very good, and some are still not perfect, like this anticodon loop here. So we will say we will conclude that predicting the unstructured modules with non-reciprocal interactions is still very difficult. And so currently, for the insights of RNA puzzles is that we have made some progress over the last decade, and also we predicted well predicted some of the rib switch and the ribozyme structures, and some RFM families have been very well predicted. And but still, we do have some bottlenecks like the non-reciprocal interaction and the large topology, and also difficult to infer the function from the structure, also dynamics of the combinational change. And so there can be some possible solutions, and so the prediction groups are actively developing new algorithms to conquer these problems. And here are some brief applications. One is in the Zika virus, sfRNA. So the Zika virus is escapes the cutting from the cellular nucleus, exonuclease, and it has a very special RNA 3D structure here. And then in RNA puzzles, we can well predict this sfRNA structure. So some of the prediction groups really did a very nice job in this prediction. And also another case is the COVID-19 structure, the untranslated region structure prediction. So this work was also in collaboration with Marta, and so we did some prediction in the 5' UTR and 3' UTR, and then we think the COVID-19 virus can be a cap-dependent translation initiation mechanism, and it could be highly related to the translation of the viral protein can be highly related to the structure of the 5' and 3' UTR. And then we predict the structures for these two ends, 5' UTR and 3' UTR, and we find some of the consensus in the prediction, and which may give insight to the structure and the function of these RNA structure, viral RNA structure. This puzzles we were organized in the March 2020, so it was early breakout in the COVID. So in perspective, we think that we still need a larger structure database to help us to understand the RNA 3D structure. Because for protein, we really have a very large structure database, but for RNA, the structure database is still not quite limited. And also, we should predict better non-canonical base pairs and non-reciprocal base pairs if to improve our prediction algorithm. And then maybe AI-based method can be play a positive role in the prediction. And we encourage the automatic prediction method. So here I would like to thank El Korestov, who started this RNA puzzle competition, and also all the cryptographers and all the structural prediction groups here. So we also welcome new prediction groups, and we welcome new cryptographers who can provide us some structures. So thank you very much for your attention, and I'm happy to take questions.
Thank you. So I don't see anything in the chat, but are there any questions? Sorry, there is something. Sorry. Okay, so can I read it, or do you want to read it? Have predictors similar to AlphaFold been applied? Sorry.
Yeah. I can read that for the first question. So, actually, there is some kind of new algorithms coming out from time to time, and there was recently there was a science paper published by Rondon from Stanford, and then they developed some of the AI-based algorithm for the RNA structure. But that was basically a AI-based geometric deep learning method for the scoring function, which can, when we generate lots of decoy or lots of predictor structures, it can rank the quality of these structures and try to find out the best model. So that's similar. That's kind of most similar algorithm similar to AlphaFold, I think. So maybe you can take a look. It's published in Science. It's called ARES. And the second question from Jiří is, what are the atom selections you calculate the model for IMSD for? Normally we use all atoms for the IMSD. Yeah. Normally,
all including hydrogens or
types of IMSD calculation. One is if we only select the phosphate or some of the backbone atoms, but normally we calculate the all atom IMSD. And the third question is that if I can ask you for some software modeling prediction of the structure. Oh, okay. Yeah, this for the software, I think the fastest web server is presented the RNA Composer. I think Marta presented that, and I think some of the good top modeling methods. I think one is FAFA2, which is in Rosetta. So Rosetta is also a powerful protein structure prediction package, and also it has FAFA2 inside. FAFA2 is the RNA structure model. So Rosetta is developed by David Baker group, and the RNA part was developed by Rejudas. And also there are some other modeling tools very easy to use, like SimRNA, which was developed by Janusz Bruszyński, and also 3D VFold. 3D VFold by Jiří Černý. So these are programs that you can use. And I would recommend the RNA Composer very fast, the SimRNA very easy to use and fast, and also FAFA2 in Rosetta. These ones are the ones I personally prefer. I cannot recommend which ones are best because any of them predicts the best in the RNA puzzles practice. So.
Okay, thank you. And maybe I would have last question. Are there any firm plans time-wise when the next round would happen?
Oh, the next round. So yeah, we are planning to do this. Actually, I'm trying to draft a paper about this predicting the structures in affine families. So we already have lots of structures. Maybe the next round can focus on the affine families puzzles. So we already have something about 7 or 8 puzzles in the regular puzzles, which focus on the affine families. And also we organize 20 different puzzles for 20 affine puzzles, which is a different practice. So we predict 20 affine structures, and then some of them have been experimentally validated. So I think we will try to combine these two, regular puzzle and affine puzzles, together as a next round. So this will be a new update.
Okay, thank you. So if there is no other, I don't see any other note in the chat, so I would proceed. Thank you, Zhichao, very much for the overview of RNA puzzles, and I hope a new one will be open soon. And okay, let's move to the last talk of the webinar, which is on tools we are developing here in Institute of Biotechnology, and Jiří Černý will show you some new tricks we can do with these structures.
So, so my name is Jiří Černý. I'm working at the Institute of Biotechnology of the Czech Academy of Sciences, and I'm going to present our web service called NATCO, as you can see. And the tool provides annotation, validation, refinement, and modeling of nucleic acids. And it assigns both DNA and RNA. It doesn't discriminate, using a consistent description of conformations. So NATCO is available from this natco.mos.org address, and the .mos.org actually stands for Database of Molecular Structures, so we have some big plans with it. We are developing something I could even call a whole ecosystem of tools and data resources under this domain. And the current public and still default version is this 3.2. But most of the things I will be showing on the 4.1. So as expected here on this Elixir Structural Bioinformatics community meeting, NATCO is supported by Czech Elixir, and it's one of the services provided by our node at the Institute of Biotechnology. So here we can also directly have a look at the fragment we are using, this dinucleotide fragment. We call it STEP, and the definition of 12 parameters we are using for defining the conformation of the nucleic acid. So it goes basically from C5 to O3 and includes also some additional parameters setting the relative position of basis. Dinucleotide is not ideal fragment, but it's basically the smallest. It's the smallest and most, because of the non-availability of data, also the biggest fragment we can basically work with. So here we can go through the tabs on top of the page as well. So home is this one. Help contains, it summarizes the basic features of the server and also some prerequisites for the analyzed or uploaded structures, like they have to follow some standards. Tutorial is very simple because it basically points to the latest ACTA-D paper describing this version, the 3.2 version in details. Table of conformers summarizes for all the conformers, it summarizes its 12 parameters which define them. It also shows the NPC, the so-called NPC, the nucleotide conformer code. It shows the numbers, like their frequencies of these conformers in DNA or RNA or END. And there is also something we call CANA, conformation alphabet of nucleic acids, which groups some parts of these NPCs into one category by similarity. And similarity can be also like user defined. So there is one NPC, but possibly more variants of the alphabet. So yeah, and the good news that's why we actually tried to lower the number of categories is that it's we have 96 conformers. So for some people it's a bit difficult to deal with them. So the next tab is browsing of conformers. It allows, like, you can choose the conformation, the data set, either non-redundant set of crystal structures or the whole EBB, include or not. For this version, it's not so good idea to include large structures because we are still using JMOL, which is slow. It's corrected in the version 4.1. About basically, this is just a list of changes, features and changes which were introduced during the time. You can obviously read the papers and download the underlying data, the torsion averages, their standard deviations, Cartesian coordinates for these like typical representation of the conformer, And there are also scripts, example scripts for programmatic access to the web service. So you can send a coordinate file or only like selected coordinates or selected torsions. And you can download the papers as well here. So from here, I'll jump to version 4.1. The initial page looks more or less the same. And on the left side, you can do two things. You can choose your own PDB or CIF formatted coordinate file and upload it for analysis. That's it. Or you can also add some experimental data, either structure factors in SFCIF or MTZ format, or CCP4 or MRC maps. All is converted and converted to format for density server, a part of MolStar, because now the service is using MolStar, and we introduced some additions which were programmed by Zhichao Miao from my lab. And these changes are mostly also propagated back to the main branch of MolStar. NATCO is internally using MMCIF, so you can upload PDB, but it is converted to CIF. And if there are some missing residues or some weird topologies, it's processed using Maxit program, generously provided by RCSB PDB, which in some cases, like if you upload model which contains too weird bond lines and so on, it complains that it doesn't recognize the chemical things. But for more or less normally behaving models, it works. So it accepts also theoretical models. So its primary purpose of this web service is to analyze experimental structures. So on the right side, you can, so if you don't have your own experimental data, you can use other experimental data. So you can type the PDB ID code and analyze structures deposited in the RCSB PDB or PDB redo with the same code. Or, as the simplest way, just click this example on DNA structure. So this is the result page, and it shows many things. So let's start here, upper right. So it shows the title of the structure, and it serves as a link to the RCSB PDB. And then there is a block of summary information about the structure, how many steps we detected, if it contained one or more models. You can directly download the results in CSV or JSON file, like the full results, not just this table, which I'll talk about in a minute. Then it kind of tells you how many steps were found in these RMSD ranges. So if everything is green, it's fine. If there are like some steps with RMSD between 0.5 and 1 angstrom or even higher, then yeah, you may be in trouble. Then it prints the overall confal for the whole structure, and confal is a score going from 0 for outliers to 100 for like perfect fit to the ideal reference of the ideal definition of the conformers, and also a percentile value with regard to the rest of the PDB. The same things are here in a graphical way in these sliders. Here you can see only green, which corresponds to these 22 well-resolved steps. In some structures, you will see a few pieces of yellow or red here as well. In some structures, you will see only yellow and red, but that's other story. So the table below gives another level of details, like really per step. So you see the step name, the conformer class, the corresponding canal letter, the confal for this step only, and the Cartesian RMSD. You can by clicking the header, you can sort the table, so you can look for representatives which are like best in terms of the torsional space, or best or worst in case in the RMSD point of view. So on the left side, you can see the actual structure, and there are some blue things on it. We call it pyramids, and they represent the conformation in graphical form. So below you can see this color code. So this light blue kind of shows you that the structure is mostly B form DNA. But there are also some dark blue corresponding to B2, and also, for some people unexpectedly, there are some pink things corresponding to A-like forms. So this the Cassandra DNA, which is by many, especially modelers, taken as the ideal B1 representative of a structure, it is not so perfect B1. And it shows that even such simple structure can be in fact quite complex. Yeah, maybe I forgot that the size of the pyramid actually corresponds to the conval. So the bigger the pyramid, like the more similar to the ideal conformation it is, and vice versa. So now we can, like, if you hover over the structure, it shows you some more detail here. It's a bit difficult to show it, but now, okay, I'll stop over, and it shows like the sequence of it. What is the conformation, its score, conval score, and RMSD. If you click it in the structure, it selects it and overlays the reference steps, and you can click a row in the table as well. The green thing overlaid shows the BB00 representative, and there is also an overlapping previous step and follow step in dark blue and cyan. This will be useful in a moment. So there, for like even next level of information, you can go into these tabs. First one is, it shows the actual values, like how different is it with respect to the reference, what are the score per parameter. And you can obviously modify also the view. So you can turn on protein hetero, like ligands, water, only water is there. You can switch between cartoon and sticks representation. You make the pyramids transparent so you can see the phosphate, which might be useful. Actually, you see that there is something unexpected here. And you can also turn them off completely. You can change their colors if you prefer. Selectively turn them on and off. And then you can also use this spheres mode, which is not so useful for small structures, but for something like this ribosome, where you barely see anything. So if we go here, we can turn off the pyramids because they are not so visible anyway. And we can turn this sphere, so you immediately see nicely the helical regions, but helical regions are basically boring, so you can turn these most often A conformers off. And now you see some red and green things spread it. So these are the interesting, usually functionally relevant open and intercalated and Z-like steps. So if you go back here, there is also something called restrain mode, which allows you to generate, as it kind of suggests, restraints, and it extends this table. And if you select an alternative conformation, like for example here, it changes. Kind of, it's indicated in red, so something is changed. So you can save it. You go for a different step, change something else, save. So you can generate few sets of restraints. You can go back, delete it, and so on. And obviously, you can download them and use them for refinement in RefMac and Penix, and also as a template for MMB macromolecule builder, which is, I could answer that, but I was waiting. This is MMB is also one of the tools which you can use for modeling RNA, DNA, and protein structures as well. So, how are you about time? Yeah, so basically. So I can go back to something I kind of skipped here, and this is this similar tab, which shows two interactive plots. Like you can zoom it, click it, whatever. I'll go to this step, or even better, I'll go here. This is like RNA example. This is tetraloop structure, and we kind of know that the tetraloop, in terms of MPC, some versions of tetraloops correspond to AA00, 1 open 03, and AA08. Okay, but here it shows 00. This plot basically shows that there is actually a good substitute. It fits both in the Cartesian and Euclidean space. So if I click it, you can immediately see that the overlap on here on the left, like this hanging green thing, kind of folds back, and it improves the compatibility of the previous and following steps. Again, you can save it as restraints, refine it, and so, and with that, I guess that's in the time we have, that's probably everything I can show about NATCO. So if you find it interesting, or even better, have some crystallographic or KREM data you would like to process and improve, please contact us. And we're also obviously open to feature suggestions, and thank you for your attention.